Hello, this is Tommy. Welcome back to Chatomics. So in today's video, I'm going to clarify some of the confusions about PCA and specifically when to use the center and the scale arguments in R's prcomp function. So while tools like threads run PCA abstract away these details, understanding what happens under the hood is crucial for proper analysis and troubleshooting. And in this post, I will show you exactly what center and scale do, why they matter, and what happens when you get them wrong. So first, we will start with a dummy example to build some intuition. Then we will dive into a real PBMC 3K single-cell RNA sequencing data sets. But before we jump into the arguments, let's quickly review what PCA does. Okay, PCA stands for Principal Component Analysis. It finds the new axis or principal components that capture the most variance in your data. Right? The first PC captured the most variance, and the second PC captured the second most. And it is orthogonal to the first, and so on. Right? So the prcomp function in R, so that's the go-to function, I think built-in function in R, base R, to do a principal component analysis. And it has two key arguments that control the data pre-processing steps, center and scale. So center means each variable will be shifted to have mean of 0. Okay, so X, for example, it's a variable and then it has multiple values, and you calculate the mean, and then you can do X minus that mean. So every variable will be shifted to have mean of 0. And scale means standardization, right? So each variable will be scaled to have the unit variance. So the default will be false. So if you type $prcomp, and you will see these two arguments. Center actually is true by default, scale is false. Okay, so when we say scale or standardization, it means X minus the mean or mu divided by the standard deviation. Right? So let's see why these two arguments matter. So let's do this with one dummy example. Okay, let's load the libraries and set the seed for reproducibility, and we're going to simulate some of the data. So we have 100 samples here, or 100 cells, for example. And then we have two features, and we have also two groups or two clusters in this data sets. So we're simulated by this random normalization, random normal distribution. So we simulate 50 of them with mean of 2, standard deviation of 0.3. And let's do that. Feature 1 and for group 1 and feature 1 for group 2. So group 1 and group 2, they both have the same feature 1, but the mean actually is different by 2, right? But the standard deviation is small. So this is a feature that can separate group 1 and group 2. Now let's simulate for feature 2 here. And now again, but we have a big mean of 1,000. And for feature 2 for group 1, and feature 2 for group 2, they have mean of 1,050. So it's 50 of differences, but the standard deviation is 200. It's much bigger. Okay, let's do that and combine them into a data matrix, and then we can add some group labels. The first 50, they are group 1, and the next 50 rows, they are group 2. Okay, so if we look at the data, it's like this. So we have 100 rows and 2 columns, feature 1, feature 2. Okay, and the first 50 rows are group 1, the second 50 rows, they are group 2. So we can actually examine the variance and the group set for each feature. Okay, we use the apply function. It goes to this data matrix, and then 2 means by column, 1 means by row. So we want to calculate the mean for each column. So that's for feature 1 and feature 2, we are calculating the mean. So we say the mean value of feature 1 is 3, mean value of the feature 2 is 1007. Okay, it's much bigger. And similarly, we can calculate the variance, right? Again, the variance for feature 2 is also much bigger than feature 1. Okay, we can check how well actually each feature separates those two groups by calculating the differences of those mean, right? So if you look at the first, the mean difference is 2 for using feature 1, group 2, minus feature 1, group 1. Okay, if you look at the second one, the differences of the feature 2 for group 1 and group 2 is 75. Looks like it's much bigger than this, but we need to calculate the effect size, taking the variance into account. So in this case, we need to divide it by the sum of the variance, divided by 2, and then the square root. So if we do this, feature 1, the effect size is 6.5, and for feature 2, because the variance is so big, and the effect size is actually only 0.416. So meaning feature 1 actually is more useful to separate these two groups. Okay? So now let's first see what happens when we skip both centering and scaling when we do principal component analysis. So we use prcomp, data matrix, and center equals to false and scale equals to false. By the way, usually prcomp accepts this data matrix. Rows are samples, columns are features. Okay, so if we do that, then we can check the loadings by access the rotation. So you can see actually for PC1, feature 2 actually is much bigger here, with the load almost 1, but for feature 1, the loading is very small here. Okay, and we can visualize it. Okay, so now we just put the principal components into this data frame for us to do ggplot plotting. If we look at this, so it's essentially just a data frame, okay, for PC1, PC2, and the last column is the group ID. So the first 50 rows are group 1, the second 50 rows are group 2. Okay, now we can use ggplot to visualize. Okay, it's like just PCA plot. Take a look. So as you can see here, so in PC1, first of all, it actually accounts for most of the variance, like 99.9%. So PC1 has always captured the most variance compared to other PCs. But in this case, because feature 2, it's much bigger in terms of mean values, without centering, it actually dominates the variance. So PC1 here, if you look at the PC1 here, those two groups, you can't differentiate them, right? Although in PC2 here, you can somehow you can actually separate them. But in this case, the variance is driven by the feature 2 here. Okay. So now let's center data, but let's still skip the scaling here. Okay. The same data matrix. Center equals true, scale equals to false, no scale. And look at the standard deviation for the P1, PC2. Again, PC1 has much more variance or standard deviation here. And let's look at the rotations, the loadings here. Again, feature 2, it's dominating the loadings here, okay? This is because without scaling, because the feature 2 also has a much bigger variance. So that's why in this case, PC1 is also driven by feature 2, okay? Let's actually visualize it, okay? Now, similar to the last code trunk, now just put the principal components into a data frame. If you look at the first 6 rows here, PC1, PC2, and then a group. But in this case, no scaling, but only centering. And in this, we will actually plot these two, the raw data also side by side. Essentially, the data matrix, the first column and the second column, those are the raw data. And now group equals groups. So because we only have 2 features here, you can visualize it in this 2D scatter plot, feature 1, feature 2. And you really don't need actually dimension reduction, but this is just a dummy example to demonstrate the effect of centering and scaling. Okay, let's do that. Now we can plot them, these 2 plots side by side. So this is just the scatter plot, feature 1, feature 2, and second is the principal component plot for PC1 and PC2, but with centering but no scaling. Okay. Now let's look at them side by side. If we look at it. So again, in the figure on the left here, this is the raw data. We only have 2 variables here. Feature 1 on the x-axis, feature 2 on the y-axis. And you can see the scale. Feature 1 is much smaller, and the feature 2 is much bigger. However, because you can clearly separate these 2 groups using feature 1 here, right? 2 groups here. But on the y-axis, if you use this feature 2, you can barely separate these 2 clusters or 2 groups. If similarly, for the PCA, in the first principal component, it's also explains the most variance here, 99.9%, but it's because it's dominated by feature 2 of its bigger variance. And although indeed in actually principal component 2, you can separate these 2 groups, but PC2 explains like 0.1% of the total variance. Okay. So kind of, I think centering kind of helped a little bit, but we still have a poor separation in PC1 because of the variance differences. So the solution here is to do both centering and the scaling. Okay, let's do it. Scale, center equals 2, scale equals to 2. Let's take a look. Standard deviation, now you have much better standard deviation or variance for those 2 principal components. If you look at the loadings for PC1, now feature 1 and feature 2, they kind of contribute equally to PC1. And well, let's visualize it. Okay, it's the same code here. If we look at the variance that I explained for each PC, no scaling and with scaling, and then put those 2 together into a same data frame. And let's take a look. This is the variance and the PCA plot side by side. Okay, so what has changed, right? So no centering, no scaling, it's a disaster, right? Groups totally overlap in principal component 1, right? If you do centering and no scaling, PC1 explains 99.9% of variance, but it doesn't separate the group well, right? It captures the noise in feature 2. And the groups are separate in PC2, which explains a little variance, okay? If we do centering and scaling like here, PC1 roughly explains 60% of the variance, and now it also clearly separates the groups. Actually, both features can contribute. Now let's put these 3 scenarios side by side and visualize them, okay? Okay, let's visualize them side by side. This is what we see here. Okay, no center, no scale. PC1, they're totally overlap with each other on PC1, although in PC2 you do actually see some separation here. If you do center only and no scaling, again in PC1, like still dominant, the variance is dominated by the feature 2, which has really big variance. But if you do actually center plus scale, now PC1 and PC2, they kind of explain similar amount of variance, and now both feature 1 and feature 2 contribute to separation of the two groups. Okay, so again, this is a dummy example. Like I think if we can actually simulate maybe a little bit a better example to show, okay, maybe those two groups, they totally actually mingled with each other, and only you do center and scale, you can separate it really well. Okay, so this is the lesson here. Okay, always center. So without centering, high mean features to create artificial variance from their origin, and scale when needed. So when features have different scales, high variance features, they will dominate even if they are just noise. Okay, so for most of the exploratory analysis, you want both centering and scaling for almost all the time. Now let's see this in action with the real single-cell RNA sequencing data. We'll use the PBMC 3K dataset from SURAT. Okay, let's load the library and fetch the data, and we need to update the SURAT object using the update SURAT object function, otherwise it gives you error. Now it's just routine standard pre-processing. Normalize it and find the variable features, most variable genes like 2000, and again scale the data using scale data function. And SURAT scale data actually centers and scales each gene. Okay, so for comparison, let's also get the normalized but not scaled data. Okay, we can extract the normalized counts, okay, for the variable genes. So get the variable features and get assay. The data layer will contain the log normalized data but no any scaling. Okay, so let's do that. And remember, prcomp, they require row samples, columns, features. So we have to transpose that matrix, okay? Because in this data, if you look at it, normalized data, rows actually are genes, columns are cells, okay? We should actually transpose it. We can also transpose the scale, the data as well, okay? If you look at the dimension, it's 2,700 cells and 2,000 genes here. Okay. Okay, let's understand what SURAT does. Okay, before we run PCA ourselves. So SURAT actually takes the scale data from scale data function, and the scale data in SURAT is already centered and scaled. Okay, if you do question mark scale data, you will see the default is both are true. So scale data, and if you look at the help, both are true here. Okay, and then SURAT run PCA on the scale data. We can verify this using the SURAT's wrapper here. And then we take out the principal components using the embedding function, and let's take a look at the first 5 columns and then first 6 rows. Okay, this is the principal components SURAT calculates. And let's just do it manually using the prcomp function. Okay, and by the way, actually for single cell data, SURAT use this LBA function for faster calculation, but prcomp gives the same results. Okay. And I have an old blog post here you can read for details. Okay, let's calculate the principal components by ourselves using the scaled data transposed. Because it's already scaled, we have to put center equals to force and scale equals to force. Okay. And then let's take a look at the first 5 principal components here, and look at the first 6 rows here. And if you compare this with what SURAT calculates, it's actually exactly the same, with only the difference of a sign differences. Because principal components, the sign doesn't matter. So essentially, those are the same results. Okay, if you look, just read carefully, they're the same number. So essentially, we recreated threads of principal component analysis using prcomp, not this low-level function. Okay. If you look at the correlation for this two, the first column of this two principal components is -1. Okay, it's perfectly correlated. They're the same. And so let's try without centering. Okay. So we will use the log normalized data, not the scaled data, okay, to properly demonstrate this. Okay, use the normalized data, log normalized. Now, this is center equals true, scale equals false, but we can do also center equals to false and scale equals to false to compare these two scenarios. Okay, and because, okay, let's do that and do that. And the normalized data, because this just log normalized, it should not actually have mean of 0. So if you take a look of that, it's calculating for each gene to look at the mean value, and then we take the mean of the mean here. So it's 0.18. This is not 0. Okay. Now we can compare these two principal, two principal component analysis with centering and without centering here. So we take a look here. This is a data frame for plotting, but we can also calculate the correlation between PC1 in both the centered and not centered analysis. If we do that, so actually the PC1 in two different analysis, they actually are still highly correlated. But if you look at the principal component 2, the correlation is only -0.13, meaning that they are capturing different patterns here. Okay, let's visualize them, put them into the same data frame again. Look at the centered and then not centered. Put them together. Let's take a look. Okay. So this is centering. This is without centering. What's the difference can you see right here? So after centering, right, PC2 now here, separate naive CD4 T cells, memory CD4 T cells, like here, NK cells, CD8 cells, and also the B cells, much better than this without centering here. Here is like a blob here. But interestingly, actually you see without centering, it seems to better separate the CD16 monocytes, FCGR3A, CD16, and also the CD14 positive monocytes. But as you will see later, if we further scale the data now, you can also separate this to CD14 and CD16 positive monocytes. Okay. Let's look at the top loadings, okay, for these two different methods. Okay, the PCA without centering, look at the rotation, and let's take a look at the first 10 genes here, and also for the centered one. So if you look at the first 10 genes loadings without centering, you see MALATA1, actin B. I know actin B is a housekeeping gene, so that's why it has actually a high expression level. MALATA1 is also highly abundant non-coding RNA, which showed up in many of the single-cell RNA sequencing data. So this really means, principal component 1 is capturing those genes like with big mean values here, right? But and those two genes, those are actually structural proteins, which means they should also have high expression values. And if you look at the loadings after you center the data, now those genes, for example, LYZ, CST3, those are myeloid monocytes markers, as 109 and A8, those are monocytes markers as well. And those three genes, those are immune cell activation markers. So, which means it's capturing more actually biological meaning or variance in your data. Okay, And we can verify this by checking the mean expression level for those genes. Okay, you can just apply, look at the gene, top genes, and look at the mean value. So let's take a look. For those genes, you have, without centering, those genes have high, the top genes, they have actually high expression value compared to the genes here. Like this actually has much smaller value than here. Okay. Again, those are the log normalized data. Okay, so a value of 3 and a value of 1, actually those are big differences. So the key insight here is that without centering, the PC1 loads on genes with high mean expression. Okay, for example, housekeeping genes that are abundant, but that doesn't tell us about the cell type differences. With centering, PC1 loads on genes with high variance between cells. So, okay, so markers that differ between cell types, and this captures biological variation. Okay, so that's why like centering is absolutely critical for principal component analysis on gene expression data. Okay, let's try PCA on the log normalized data with centering and also scaling, but we should compare without or with a scaling. Okay. Okay, the scaled, this is already both scaled and centered, and normalized here is only centered but not scaled. Okay, we can calculate the variance that explained. If we do this and plot them, as you can see here, with the scaled data, centered and scaled, the variance is more actually evenly distributed, okay, by different principal components. However, if you don't scale it, only center it, the first two principal components, they actually explain a very big variance, okay? But is this biological signal or just the high variance genes that are dominating the first two PCs, for example? Let's take a look here. So this is the data frame to compare those two principal components. Analysis here, okay, that's scaled and normalized. So essentially, this is only centered but not scaled. This is centered and scaled. And let's take a look at them side by side. So if you look at this, actually this, if only centered data, it still separates the cell types in a kind of relatively well. But if you do both centering and scaling, now you can further actually separate the CD14 and CD16 monocytes. Okay, suggesting that actually doing centering and scaling can help to separate the cell types, and this is usually good for our data analysis. Okay, the standard practice in single-cell RNA sequencing is to scale the data first, okay, because it gives each gene equal weight, and it's also more robust to outliers. Okay, so the key takeaways is here. Okay, when to use center equals true. So you almost always want to center the data. And then when to use scale equals true, right? So when the features are on different scales, for example, mixing counts and the ratios, like those counts like could be 1 to 1,000, but ratios will be like 0 to 1, right? The scales are very different. And if you want each feature to contribute equally regardless of its variance, so you want scale equals true as well. So don't use scale equals true because if the data is already scaled, like Surratt's scale data, if it's already scaled, you don't need to scale it again. Okay? So for single-cell RNA sequencing analysis specifically, it's always log normalize the counts, okay, by the library size and then log transform it. And then you scale the various features, center and scales, okay, both. And then you run PCA using the center equals true and the scale equals false. Actually, center equals false because after the scaled data from in Surratt, it's actually both center and scale all the genes, the most variable genes, okay? Okay, so why does actually SRED scale genes? Okay, you might wonder, okay, if you are selecting only highly variable genes, why scale them? Won't that remove the variance information that we just selected for? So the answer is that we select highly variable genes to focus on genes that vary across cells, biological signal, but we scale them so that the high mean genes, they don't dominate PCA, and each gene contributes equally to the principal components. So we capture the pattern of co-expression, not just the expression magnitude, okay? So we select genes that vary a lot, which has biological meaning, and then we scale them so we can focus on how they co-vary rather than how much they vary. This is actually very similar to making a heat map when you want to scale it, and then you want to find the co-variant genes, so the genes with the same pattern that are up in certain cells and down in other cells, for example. So always center your data before PCA and scale when features are on different scales. And for single-cell RNA sequencing analysis, it's always scale genes first, and then PCA without scaling, okay? So understanding the arguments helps you troubleshoot and understand what tools like SRED are doing. And I hope this video demystified prcomp for you. And yeah, happy learning. And that's it for today, and I will see you next time. And make sure you subscribe to ChatRMix and share it with your friends if you find it helpful.